Last time we talked about how to build a virus particle like this, the icosahedral symmetry of polio. Today we're going to talk about how to take it apart to get the genome out. And you see this model, I have a hole at one end. Is that really what happens? Or is there something else? So we'll find out today. Not just for viruses like this that have icosahedral shells, but for viruses with membranes, the two general categories of getting the nucleic acid into cells. So we're going to talk about attachment and entry. Viruses, of course, are obligate intracellular parasites. We talked about that in the first lecture. They have to get inside of a cell to carry out their replication cycle. But they're too big to diffuse across the plasma membrane. Only small molecules of certain kinds can do that. So there has to be a specific way for them to get into the cell. And this is all about finding a cell surface receptor. The first part is finding the right cell. Viruses go past lots of cells in their travels. They have to find the right one. And there's a lot of adherence via electrostatics on various cells that has no specificity. Viruses bump into cells as they go through your body, as they land on you, and so forth. And if it happens that that cell has a receptor for the virus will attach to it and initiate entry. So receptors are needed to get viruses into cells for the most part. Sometimes there's more than one receptor molecule as well, and then the genome gets inside the cell. And that's what we're going to be talking about today. So a virus receptor is needed for all the viruses we talk about, with the exception of viruses of fungi and viruses of plants. Viruses of fungi apparently never leave the cell. It's very interesting. They stay within the cytoplasm where they replicate, and then when the cell divides, the virus goes to the next cell. Viruses of plants do have extracellular phases. They get out of plant cells, but they get into cells by mechanical damage, either by an insect or a nematode or farm equipment damaging the plant. This introduces viruses into plant cells. There are no receptors. But for animal cells and bacterial cells, there are receptors for viruses. That's what we'll talk about today. This is a relatively new field. As recently as 1985, only one virus receptor was known, and that is sialic acid for influenza virus. So when I talk about a virus receptor, what I mean is a molecule on the cell surface that the virus is attached to. So in traditional parlance, the virus is the ligand, the cell has the receptor. Okay, sometimes people get it the other way around. The virus has the receptor and the ligand is not on the cell. That's not how I do it. Cell has the receptor, the virus is the ligand. Here are some examples of cell surface receptors for viruses. Now at the top is a model of the plasma membrane of a mammalian cell, and you probably know that it is a lipid bilayer composed of polar phospholipids. And within that bilayer are a variety of different kinds of proteins. There are integral membrane proteins that cross the membrane, and they have extracellular and intracellular domains on them. You have lipid-anchored proteins that may be on one side of the membrane or the other. They typically don't traverse the membrane. And many proteins that are in the membrane are glycosylated. Those are these yellow spheres and chains of them here. These are chains of sugar molecules that are attached to proteins. They have a variety of functions. We call, of course, the proteins that have them glycoproteins. So on the bottom are just some examples of receptors for viruses. You can see a broad array of cell surface molecules. They have transmembrane domains. Sometimes there's a single receptor for a virus. Sometimes a virus needs two, as we'll see. In some cases, the receptor's a dimer, as shown here for the integrin. And sometimes the receptors are quite simple, like this ganglioside for a GM1, which is simply a glycolipid and not actually a protein embedded in the membrane. So viruses attach to a wide range of cell surface molecules. There doesn't seem to be any particular type of cell surface protein or glycoprotein that is bound by virus receptors. In this slide, you can see they're labeled the viruses that bind to these various receptors. For example, vaccinia virus, measles virus, SV40, rhinovirus, foot and mouth disease virus, HIV, which requires two receptors to get into a cell, CD4 and a chemokine receptor, and adenoviruses. And we'll talk about some of these and how they work today. Now, it's not always one-on-one. It's not always one virus, one receptor. There are variations on the theme. Different viruses sometimes bind the same receptor. So, for example, adenovirus, which we've talked a little bit about, and a virus we haven't talked about, Coxsackie virus, have a common primary receptor. They both bind to the same cell surface molecule. Coxsackie virus, as you might guess, is named after Coxsackie, New York, exit 21B on the New York State Thruway. The virus was isolated there in the 50s, and it was named after the town. And I took that picture driving by, holding the camera in one hand. I always wanted a picture to show you guys in my lectures, and there it is. Swine herpes virus, called pseudorabies virus, binds the same receptor as poliovirus. Again, two different viruses binding the same receptor. Also, viruses of the same family could bind different receptors. So rhinoviruses are all within a genus of the same virus family. There are 3 different rhinovirus receptors. Retroviruses, which are all in the same family, can bind to 16 different receptors. I don't expect you to remember any of these receptors. I just want you to know that there's lots of variation in what a virus binds. But the virus usually binds whatever receptor that it initially is bound to. So poliovirus binds one receptor. That doesn't change. The retroviruses bind 16 different receptors, and that doesn't change. Did you have a question? That's a good question. So why would different viruses bind the same receptor? Probably it's not because of similar structures. Adenoviruses and Coxsackie viruses are quite different, as are herpes viruses and poliovirus. So herpes virus is a glycoprotein binding the receptor. Polio, it's an icosahedral capsid. So it must be a very small domain in the particle that is recognizing the receptor, but we don't know the answer to that. Viruses of the same family can bind different receptors. I just told you retroviruses bind 16 different receptors. There's a repetition of that. And here's an example of one virus that can bind multiple receptors. This is herpes simplex virus type 1. And it has a variety of glycoproteins embedded in its lipid envelope. And they're labeled here GP42, GB, etc. And they bind different cell surface receptors. The cell surface receptors are shown at the bottom. For example, integrins, nectin-1, HVEM, etc. So receptor interaction of viruses can range from the very simple, one virus, one receptor, to the more complex, multiple viruses binding a single receptor, or viruses binding many receptors. So let's look at a couple of examples of the topology of receptor binding. Now, we have a lot of structural data on viruses and how they interact with their cellular receptors. On the upper left is a cryo-EM structure of poliovirus with its receptor attached to it. So the virus you can see in 3 colors, blue, yellow, and red. And it's very much like the structure that I have here on the front desk of poliovirus. This structure is without the receptor. But in that structure, the virus was mixed with a soluble form of the receptor, that is, without the membrane, and the cryo-EM structure was determined. And what you can see is that a lot of receptors can bind the virus particle. Now, icosahedral symmetry predicts that there are 60 copies of a binding site for a receptor. And in fact, you can count 60 molecules on these reconstructions. And you can see exactly where they bind. For example, here is a 5-fold axis of symmetry at the center of that blue pentamer, if you will. And you can see there are 1, 2, 3, 4, 5 poliovirus receptors binding around that 5-fold axis. We actually think that only a few receptors are needed to be bound by the virus in order for the virus to enter cells. We don't think you have to occupy every receptor binding site on the particle. We also know what part of the receptor dips into the canyon because people have done higher resolution structures of the receptor. That's shown in the middle here. So here you can see the alpha carbon trace of the receptor because it's been solved at higher resolution than the cryo-EM. And this can actually be fitted into the cryo-EM structure so that we know which part of the receptor is fitting into the virus particle. Now, For poliovirus, the receptor dips into a little depression on the surface. It's schematized on the lower left here. We call it a canyon. And the receptor CD155 is shown here in the cell membrane. And it dips into a little depression in the surface, and that's how the virus attaches. But it's not always the case. You know, this lock and key metaphor that we like to use in biology certainly is not always the case for virus receptor interactions. And that's illustrated on the upper right. This is rhinovirus bound to one of the three rhinovirus receptors, which happens to be low-density lipoprotein receptor. So this is a receptor in us that is involved in cholesterol transport, and the virus has evolved to use it. And the receptor in this model of rhinovirus is shown in gray. And you can see it's not fitting around the canyon. So there in the middle is a five-fold axis of symmetry. You can tell because there are 5 receptors bound around it. And here the receptors are actually binding on this plateau at the five-fold axis. So if you look at the polio structure on the lower left, that line shows you the five-fold axis of symmetry. And there is a flat surface at the top, which we call the plateau. And that is actually in rhinovirus where the receptor is binding. So even though rhinoviruses do have a canyon, in some cases the receptor binds in these flat regions. And again, these are non-covalent interactions. They're simply electrostatic protein-protein interactions. They have to be broken at some point, so they can't be covalent. This reminds me, the fact that this is low-density lipoprotein receptor binding rhino. This reminds me to tell you that virus receptors do not exist so that viruses can get into cells, okay? It's not the only thing that they do. Every virus receptor that we know of has a cell function. And so the evolution of viruses is such that they have evolved over the years to be able to recognize cell surface proteins. So if you did think that maybe these proteins are there just for the virus, wipe that from your thoughts. And if that were the case, of course, we could target them with ligands and block attachment. We can't do that because the receptors have their own cellular functions, and we would disrupt those if we did that. So that's an example of an icosahedral particle, how it would bind to a receptor. Here's another icosahedral particle, adenovirus, where the strategy is slightly different. Now, adenovirus, you remember in this electron micrograph on the right, built with icosahedral symmetry, but it has extra proteins that make these fibers at each five-fold axis of symmetry. And it is the very tip of the fiber, called the knob, that binds the cellular receptor. So on the left is a high-resolution structure of the knob and the shaft and the penton base. So the penton base is the structure, the structural unit, or the capsomer at the five-fold axis of symmetry. And then the rest of them are hexamers, called hexons, in fact. And then we have a shaft, which is a trimer. And then at the very top, there's a knob. And the knob is what interacts with the cellular receptor. And here in the middle, we have a cartoon of that. Here's the knob of an adenovirus. And in the structure in panel B, the red and blue and yellow is the knob of the adenovirus fitting down into the receptor for the virus, which is shown in light blue. The receptor is shown as a trimer, and the trimeric knob is fitting into it. So in the case of adenovirus, protein-protein interactions between the knob, which is at the end of this very long shaft, and the cellular receptor. So those are two examples of icosahedral viruses. Now, the other class of viruses has membranes around them. So how do they attach to cellular receptors? Well, as we learned maybe last time, when viruses have envelopes, which are shown here as the gray line for this influenza virus, they must also have viral glycoproteins in the envelope. Otherwise, the viruses wouldn't be able to attach to cells. So influenza virus has a number of different glycoproteins in its envelope. And the important one for today is the one called the hemagglutinin, or HA. We'll be talking about this molecule a lot. It's one of our model viruses, and the HA has a lot of functions. And that's shown in purple color here. And in this figure, you can see how the HA is engaging with a cellular receptor. So here's the cell membrane, also a gray lipid or a brown lipid bilayer, and multiple HAs interacting with the receptor. This is actually a cartoon and does not really represent what's going on. So let's take a closer molecular look at how flu HA interacts with its cellular receptor. There are a couple of different receptors for influenza virus, and they're all sugar molecules. They're sugar molecules that are attached to proteins. And the nature of the protein doesn't matter. What matters is the very last sugar on the chains of glycoproteins. So here on the left is a transmembrane glycoprotein on a cell. So you can see the protein is in cyan. And then we have two chains of sugars attached to it. So this is a typical glycoprotein. You have multiple subunits of sugars attached to it. And influenza viruses bind to sialic acid, which is typically the very last sugar on a sugar chain on a glycoprotein. So that a red arrow shows you where sialic acid would be. Sialic acids come in different flavors, primarily depending on how they're attached to the next sugar in the chain. Now, influenza viruses, as we'll learn later, bind to different kinds of sialic acids. The influenza A and B viruses, which are the ones that cause most of our infections, our vaccine against flu contains A and B components, they bind to N-acetyl-neurominic acid. And those are the neurominic acids shown up here. We'll get back to those in a moment. The reason they're called N-acetyl, because on the amino they have an acetyl group. The influenza C viruses, which infect many people but don't cause much disease, bind to 9-O-acetylated N-neurominic acid, which isn't shown here, and we won't talk about that further. So let's go back to the N-acetyl-neurominic acid. That's just another word for sialic acid, all right? N-acetyl-neurominic acid equals sialic acid. So you can see on the top, here's the sialic acid in yellow. It's a typical 6-carbon sugar molecule with various side groups. And you can see the next sugar in this chain is galactose. It can be a variety of other sugars. But the sialic acid is attached to it by an alpha 2,3 linkage. The 2 and the 3 simply represent the carbons in this 6-carbon ring. So that is alpha 2,3 sialic acid. The one on the bottom is alpha 2,6. The 2 carbon is connected to the 6 carbon right there by an ester bond. All right, so human influenza viruses prefer to bind to alpha 2,6 linked sialic acids, whereas avian strains prefer to bind to alpha 2,3. Now, why am I telling you this? We are very worried about avian influenza viruses, especially the ones that are very pathogenic and wipe out chicken flocks worldwide. They bind alpha 2,3 sialic acids, and they don't bind human respiratory tract tissue as well, because as you'll see later, the alpha 2 sialic acids that we have in our tract are way down in the depths of the lung, and these viruses can't reach them very readily. But we're worried that they may evolve to utilize alpha 2,6 linked sialic acid. So later when we talk about pathogenesis, we'll get back to this important point. So that's how the virus attaches to it. And here's a X-ray structure of the viral HA, the HA molecule attaching to sialic acid. So on the left is the X-ray structure of a single HA molecule. Here it's embedded in a virus membrane, and it consists of a fibrous stem and a globular head. And that's why in the cartoons we draw it that way. It's based on the crystal structure. And at the top of the globular head, in this groove, which you can just barely see at the top, that is where the sialic acid fits in. And that's expanded on the right here, and the blue is are the atoms from the HA molecule, and the yellow-greenish molecule is the sialic acid. You can see it's fitting in this little pocket here, and it's making a variety of hydrogen bonds and other interactions with the side chains of the hemagglutinin. So that is the molecular basis for HA binding to sialic acid. The sialic acid actually fits into a pocket in the very tip of the HA molecule. HIV is another virus with an envelope. It has a glycoprotein in the envelope that also attaches to cellular receptors. Here's the virus on the left. Envelope virus glycoproteins are shown as trimers. Here is the X-ray crystal structure of this glycoprotein. The brown is the cellular receptor called CD4. And the viral glycoprotein is shown in red, bound to the cellular receptor. So CD4 on the right, there's the cartoon, is brown. This is the cellular receptor, one of the 2 cellular receptors. And the part of the glycoprotein that binds CD4, it's called SU for surface. It's binding to CD4. And there's a very important cavity for that interaction, which involves a phenylalanine residue on CD4 that fits into a pocket on the glycoprotein. Very much like sialic acid fits into the flu HA, except here it's a protein fitting into the glycoprotein of HIV. So those are ways that icosahedral and enveloped viruses can attach to cellular receptors. All right, our first question is, viral receptors on the cell surface, A, can bind directly to icosahedral virus capsid proteins, B, interact with glycoproteins of envelope viruses, C, can be carbohydrate or protein molecules, D, have cellular functions, or all of the above. Which one is correct? Most of you got all of the above, which is right. They can bind directly to icosahedral virus capsid proteins. We talked about that with polio and rhino. They can interact with glycoproteins. The receptor can be carbohydrates, sialic acid, or proteins. Of course, they have cellular functions, so it's all of the above. All right, so that's how viruses attach to cells. How do they get inside of the cell? Because the genome has to get out. We can't just attach, the genome has to get out. Well, cells, of course, have processes for taking up materials that they need. On the left is one called phagocytosis. This is a process by which cells like macrophages take up big particles. You can see 1 to 2 microns in size, and basically parts of the plasma membrane come out and surround these particles. We think this may occur for some of the bigger viruses, but we're not sure. Most of the entry we'll talk about today involves the processes shown on the right, and they are called endocytosis, and they consist of what we call macropinocytosis, where the plasma membrane undergoes a ruffling, and projections come out and grab components that are outside and bring them into vesicles, and these would be molecules that the cell requires, for example. And then there's receptor-mediated endocytosis, where larger ligands have receptors for them on the cell surface, which are taken up into vesicles by the endocytic pathway. So viruses tap into both macropinocytosis and receptor-mediated endocytosis. And I think you can guess now that the receptors we've been talking about for viruses would fall into this category. The various receptors for viruses normally bind a ligand in their normal function, but viruses can bind them and be taken into the cells as well. Now, the cytoplasm is a very crowded place. And in the old days, we used to draw a cell with an empty cytoplasm and a ribosome here, the nucleus. It almost seemed as if it were empty. But this is an artist's rendition of a cell, which is probably accurate. And it's in separate panels because it's too big to put in one. And here's the plasma membrane with glycoproteins. He's drawn them looking like plants, but they're glycoproteins. We have the plasma membrane in yellow. Then you have all the actin filaments below this plasma membrane. So that's quite a barrier in itself for viruses to get in. And many viruses trigger disruption of this actin network. And then as we move further into the cell, you can see there are a variety of microtubules in here. There are ribosomes. Those are the purple ones. Here's some clathrin-coated pits, endoplasmic reticulum. And we're moving closer and closer. Now here's a nuclear pore, this big complex here, because this is the nuclear membrane. Here's the nucleus with nucleosomed DNA in it. And the nuclear pore, of course, allows things to get transported into the cell nucleus. And finally here, we're in the nucleus with all the DNA wrapped up in chromatin. The point here is that virus particles, once they bind the cell surface, they get into the cell, but they don't just diffuse throughout the cell. All right? It would take them forever. The cell is a very crowded place, it's very viscous. It would take too long for viruses to diffuse. They use active transport mechanisms that exist in cells to transport material through the actin filaments and along the microtubules. And that's illustrated on this slide. This is kind of a summary of all the entry pathways we're going to talk about. And here, for example, is a virus particle being taken up into an endosome. Many viruses bind their receptors, and the receptor-bound virus gets taken up into a clathrin-coated vesicle, and that moves to the center of the cell by binding to a microtubule, and then there are, of course, motors that bring the vesicles toward the nucleus. So they're dynamin and kinesin motors that bring the vesicles in different directions. And that's the case for all of this virus transport, whether viruses enter by clathrin-dependent endocytosis, by clathrin and caveolin independent mechanisms, by caveolin-dependent mechanisms. The vesicles travel along these microtubule networks. And as you know, the microtubules all end at the centrosome, which is quite close to the nucleus. Now here is a virus particle that's not being taken into the cell by endocytosis. This is an envelope virus, and its membrane fuses at the plasma membrane, liberating the capsid into the cell. Now this is a virus with DNA genome, so the DNA needs to get in the nucleus. So what does it do? It binds to a motor on the microtubule and gets motored down to the centrosome and then over to the nucleus where it docks onto the nuclear pore. And again, other virus genomes fuse at the, other viruses fuse at the cell surface and deposit their genomes there, and some of them don't have to move very far for them to be active. But the important point is that there's no diffusion, because that's too inefficient. There's active directed motoring, which we're starting to learn more and more about, of components, virus particles, and vesicles containing viruses into the cell. So here's a movie that someone made to illustrate a motor on a microtubule bringing a big vesicle, could have a virus in it or could have cellular components, from the plasma membrane into the cell. And this is a very nice depiction, I think, of what's going on. I think it's a little bit uncrowded, so there's too much space in here, but of course it would distract from the original idea. So again, this is a microtubule filament. This is one of the motor proteins that we know of that is an ATP-dependent motor that brings vesicles containing viruses or even virus particles into the nucleus, into the cell. Now I mentioned that some viruses don't have to travel very far within the cell because they fuse at the plasma membrane. So some viruses that are enveloped can in fact fuse at the plasma membrane. That's shown at the top here. Here is a measles virus, basically a paramyxovirus binding its receptor. The virus membrane fuses with that of the cell, and you can see as a result the nucleocapsid, that's the RNA protein complex of the virus, is deposited into the cytoplasm. Now it could stay there and probably make mRNA and make proteins. If it had to move deeper into the cell, it would certainly move by microtubule-dependent transport mechanisms. How does this fusion occur? So this is a theme we're going to cover for a little bit here. We're going to first talk about how membranes of viruses fuse with that of the cell to release the genome in a process that's shown here. So there are two mechanisms shown here. One is in panel B. And what we're looking at is a close-up of the virus membrane, and there are two viral glycoproteins here called F and HN. And these happen to be glycoproteins found in paramyxoviruses like measles virus. And these allow the virus to fuse at the plasma membrane at neutral pH, because of course the plasma membrane is neutral pH. The F protein is also the fusion protein. There is actually a fusion peptide buried within the F protein that has to be exposed by cleavage of this precursor by proteases in the cell. Now what happens here is these viruses attach to a cell receptor, which is depicted on the top panel in red, and that binding to the cell receptor induces a conformational change in the F protein, which exposes the fusion peptide. It can then insert into the membrane and catalyze fusion. So in general, the viral fusion that we're going to be talking about is catalyzed by fusion peptides that are embedded in viral glycoproteins. And they typically have to be hidden. So this fusion peptide is hidden by having it folded up against the virus surface. Why do you have to hide it? Well, if the fusion protein were just sticking out, the virus would be fusing with every membrane it encountered, and that wouldn't be good. We need specificity in the fusion reaction. And in this case, the specificity is achieved by hiding the fusion peptide and exposing it only when the viral HN glycoprotein binds the right receptor. So if it binds the wrong receptor, it's not going to induce this conformational change. So compare the first panel where the fusion peptide is hidden to the last where now the fusion peptide is inserted into the cell membrane. Now how fusion occurs we'll learn about in a few slides. So that's the paramyxovirus measles. Now HIV enters by a similar mechanism at the cell surface, and how HIV does that is shown at the bottom. And the reason I'll show you in another example is because here's an example where the virus requires two cellular receptors in order to uncoat its genome. So here on the left in panel C, we have the viral membrane. We're looking in close at one of the viral glycoproteins. And again, there is a fusion peptide. It's in part of a protein called TM in this case, and it's folded up against the virus membrane so it doesn't spontaneously fuse with any membrane it sees. Next to it is the attachment domain called SU. It's shown in yellow. So SU initially binds to the CD4 receptor on the host cell, but that's not enough to get virus infection. You need to bind to a second receptor, which is a chemokine receptor called CCR. The binding of CD4 to the viral SU protein induces a conformational change in SU, which then tickles CD4, makes for high affinity binding, and that unfolds the fusion peptide. If you look at the middle versus the right panel, you see the binding of SU to CCR makes the fusion peptide expose, it inserts into the cell membrane, and that catalyzes fusion. So we have just two examples. It's really a similar process. It's receptor-dependent exposure of the fusion peptide, except with one virus we just need one receptor interaction. With HIV we need two. So here's a movie of HIV infecting a cell, which I think really makes this clear for you. There's the blue cell is a lymphocyte in the blood, and the little particles, which you'll see, well, you can't see them yet. We're zooming in on the lymphocyte, and here are the two receptors for HIV, the CD4 in blue and the chemokine receptor in yellow. And in a moment, the virus will approach over the horizon. There's the virus, HIV, and it has the glycoproteins in the surface. You see there aren't a lot of them. The HIV glycoproteins are not very densely inserted into the virus, as is that of influenza. So there's, again, the SU portion, and the part that contains the fusion peptide is in blue. So the SU is going to bind to CD4 right there. I don't know why it's swinging, the artist's license. Then it binds with high affinity to the chemokine receptor. And that then, they take away everything so you can see it clearly, but it doesn't go away in real life. And then the fusion peptide swings out and inserts into the cell membrane. And eventually the virus in the cell membrane will get drawn very close together. We'll talk about that mechanism in a moment. And the two will fuse. Okay, so again, if you look back in this movie, initially the fusion peptide is up by the virus, so it doesn't just fuse with anything. Now here's another movie that just shows you the protein that contains the fusion peptide. So the viral membrane is down here, cell membrane is up here. And that's a monomer. In the virus, it's actually a dimer, as you could see. I think we're going to get an animation of this. Yes. So that's what happens. This is the fusion conformation with the fusion peptide up here. And this is the extended conformation, of course. And it's usually folded before the virus attaches to the cell receptor. So in this conformation, the fusion peptide up here inserted into the cell membrane, that's how fusion occurs. And you might want to go back and look at this on your own to get a sense of it. Now, once the fusion peptide is inserted into the cell membrane, the molecule undergoes what we call hairpinning. It starts to fold on itself, which brings the two membranes together. And when you bring membranes very close, you get rid of the water in between them, they begin to fuse. Okay, so the fusion occurs when the leaflets are brought together and a hole in the membrane. All right, so initially you have an extended fusion protein, it hairpins, it brings itself back down, and it turns and makes a fusion pore like this. And there's the complete pore made. Both leaflets of the layers, of the bilayers, have fused, and now you have a pore through which the viral nucleic acid can flow. I recommend looking at that by yourself several times, and you get the hang of it. All right, the next question is, which of the following does not play a role in virus entry? Clathrin-mediated endocytosis, fusion of viral and plasma membranes, diffusion of virus particles in the cytoplasm, microtubule-mediated transport, or lysosomes, which does not play a role in virus entry. All right, so this is very interesting because I gave the same lecture to medical students this morning, and I got the same result. 55% of you got C, diffusion of virus particles. And that's correct. That's the correct answer. That's what I've emphasized. You never get diffusion. But a lot of you said lysosomes. And I guess since we haven't talked about them, I should get rid of them from this question and put something else in that's correct. You'll see later on that for most viruses, they want to escape the endocytic pathway before they encounter lysosomes, otherwise they would digest the particles. But as always, there are some viruses that actually need the lysosomes, so they are part of virus entry. But I understand why you got it wrong, because I haven't taught it to you. All right, let's look at this fusion reaction for influenza virus. Here we know the most about it. We've got molecular structures, and we've characterized every step of the interaction. And a lot of these things we've already touched on. You're just going to get it in a little more detail. So we start on the upper left. An influenza virus has bound to the cell surface via HA, interacting with a sialic acid receptor. The particles being taken up into an endosome, which pinches off into a vesicle. Now, as you may know, as endosomes move from the plasma membrane towards the nucleus, they acidify. The pH drops, and that's because there are proton pumps in the endosome membrane that pump protons into the interior. The pH goes from 7 to 6.5 to 6 to 5.5 to 5. And around pH 5, the influenza virus HA undergoes a conformational change that leads to fusion of the viral and cellular membranes and release of the viral nucleocapsid. Now, how does this happen in molecular detail? Here we have 2 trimers of the HA. The HA is a trimer of 3 subunits. And here's the globular head binding to the cell receptor. As the pH drops, the protein starts to undergo a conformational change. You can see that in the second panel. Now, the fusion peptide of the HA is down here, that purple segment near the membrane. So you see there's a yellow piece of protein in the purple segment. And as the pH drops, that fusion peptide flips up, and it's shown in the middle panel, panel 3, embedded in the membrane. And there are 3 fusion peptides because it's a trimer, 1, 2, 3. So it's a pH-catalyzed rearrangement of the fusion peptide. Remember, in the other 2 viruses that were fusing at the cell surface, it's neutral pH, so it's catalyzed by interaction with a receptor. Here, it's pH-catalyzed. Now we have the fusion peptide up there. The HA molecule is now in an extended conformation, which begins to hairpin. It folds on itself, much like the GP41 of HIV did. So here's an early step in that hairpinning. All that means is that the molecule is bending in two, and when that happens, you're bringing the part of the cell membrane with the fusion peptides embedded into it very close to the viral membrane. And when you bring membranes right next to each other, so there's no water in between, they spontaneously fuse. So this hairpinning brings the two the two membranes together, and now you have a pore formed between the cellular membrane and the virus membrane. So there on the bottom in molecular detail, and on the top, that's the big picture, and the nucleocapsid gets out. Nucleocapsid, of course, is RNA plus protein, and for flu, it's really unusual. It needs to go in the nucleus. So it goes through the nuclear pore where it commences to replicate. So here is a schematic of the HA undergoing these changes, so you can see them on their own. Here on the left is the neutral pH form of the HA, shown as a monomer. Here's the fibrous neck and the globular head with the sialic acid binding site. Now, the fusion protein is down here, all the way near the membrane, and it has to be actually liberated by a by a cleavage event. So as the HA is made in the cell, the fusion peptide is not exposed. It has to be cleaved by the right cellular protease. And that's why flu only replicates in cells of our lung, because those are the only ones that have the protease that can cleave this. The cleavage exposes the N-terminus, which is shown here in yellow, and that's the fusion peptide. And so under low pH catalyzed fusion, which is the last structure, this fusion peptide pops to the top of this alpha helix and inserts into the cell membrane. So the fusion peptide is hidden, it's exposed by cleavage, and then put into the cell membrane by low pH. There are a lot of viral proteins that have a similar mechanism for fusion. They're called class 1 fusion proteins. They're typically perpendicular to the virus membrane, so we call them spikes. They're mostly alpha helical, as you can see in all these different viral structures, and they form trimers, like the HA of flu. So we have flu, a paramyxovirus, Ebolavirus, HIV, a retrovirus of mice. They all have these type 1 fusion proteins that are trimers, and they all fuse by the mechanism that I've just told you about. So here's a movie of influenza virus entry. There's the virus particle binding to the cell surface. It's taken into a clathrin-coated vesicle, which then pinches off. Now it's going into the cell. There are no microtubules here, but there should be, right, because this is moving along the microtubule network. So now the virus is in that shell. Now we have a microtubule, very good. Must have taken my course. Clathrin is coming off. We know that happens. Here we have the endosome containing the virus. And now influenza virus has popped out, and there's the ribonucleoprotein. So inside has been the low pH and the fusion that liberates the nucleoprotein, and now it's going into the nuclear pore. There are other kinds of fusion proteins on other viruses. One that I want to tell you about is called class 3. And these are distinct because they're mostly beta sheet compared to the class 1. They form dimers instead of trimers, and they're parallel to the membrane. So here's a flavivirus, like Zika virus or yellow fever virus. And you can see instead of the spikes sticking up as they do in flu, they're lying flat on the membrane. And how does this work to induce fusion? Same thing as for flu. You have the proteins lying flat on the membrane. When the virus binds its receptor and is taken into the cell, the endosome acidifies. The protein undergoes a conformational shift that puts the fusion peptide in the cell membrane. So the yellow part contains the fusion peptide. These proteins then hairpin, just like the flu HA does, brings the virus and the cell membrane together to get fusion and a fusion pore. So the mechanism exactly the same. The difference is that the protein is different and it's lying flat on the membrane, but that's not an obstacle. So here's a movie of dengue virus entry, which is a flavivirus taken into the cell by endocytosis. There's the endocytic vesicle moving on a microtubule. pH is dropping inside. We're going to look inside and watch those fusion proteins, which are flat on the virus. They're popping up, and they're going to insert into the endosome membrane. There they are inserting into the endosome membrane. So these were previously flat, the fusion peptides going up there. Now they're going to hairpin and draw the cell membrane, which is at the top, down to the viral membrane, which is on the bottom. Bring very close, and as soon as the membranes touch, getting rid of all the water in between, they're going to fuse. And then the dengue nucleic acid will come out through the pore. And that's shown on this slide. That's shown at the end of this movie. There it is coming out. And this is a plus-stranded RNA, so it can be immediately translated in the cytoplasm. Some good animations out there. Now there's an unusual twist that we learned recently with Ebolavirus entry, which is an envelope virus, filamentous of course, with a negative-stranded genome. The difference is that these viruses also fuse in an endosome, but there's a fusion receptor in the endosome. It's not just low pH mediated. So, for example, these viruses bind a cell surface receptor. They're taken into the endocytic pathway, shown here. The viral glycoprotein, as it's present in the virus, needs to be cleaved in the endosome by cellular enzymes to expose a binding site for an endosomal protein called NPC1. And when the virus glycoprotein binds to NPC1, that catalyzes fusion, and so the viral RNA can get out of the endosome into the cytoplasm. Fusion is mediated by an endosomal receptor, but it's regulated because the receptor is only in the endosome, it's not on the cell surface. NPC1 stands for Niemann-Pick protein. This is a disease of cholesterol transport. NPC1 is involved in transporting cholesterol in and out of the vesicle. And there are people who are born with defective genes for NPC1. They don't live very long, Niemann-Pick disease. And they cannot transport cholesterol, which is why they die at a young age. But their cells are also not infectable by Ebolavirus because they don't have NPC1 in the endosome. It's a little consolation to them because they die at 10 or 11 years of age, and they're not likely to be infected anyway. But their cells were really important for understanding the receptor for Ebolavirus. So I want you to remember, fusion is regulated. It can't occur in the wrong place. It can only occur at the specific place on the right cell for the virus to get in. So at the plasma membrane, we talked about neutral pH fusion, where a second protein interaction lets the fusion protein be exposed. We talked about low pH fusion, where you have proteolytic cleavage to activate the fusion protein. That happens in class 1, or sometimes for class 2 fusion proteins, you have cleavage of a second protein to liberate the fusion peptide. We didn't talk about that, but it's a different aspect. Or you can have an endosomal fusion receptor. So that's the really important point here, that fusion is regulated either at the cell surface or within the endosome by different mechanisms. All right, this question, viral fusion peptides are exposed for insertion into the host cell membrane when A, the virus particle is near a cell, B, the virus particle is in the cytoplasm, C, trimers of the fusion peptides form, D, the endosome becomes acidified, E, the virus is docked on the nuclear pore. So when are the fusion peptides exposed for insertion? All right, so I predict that the answer is going to be all over the place. Let's see. Yeah, sort of, right? So 55% of you said when the endosome becomes acidified. Right. So the acidification exposes the fusion peptide. So when a virus particle is near a cell, a lot of you pick that. That's not enough, right? Because if it's not when it's engaging a receptor, it shouldn't be fusing with the wrong cell. So the requirement for a receptor is important. So it can't just be near a cell. Yeah?
How is there an endosome if it hasn't fused, if it hasn't inserted into the cell membrane yet?
Well, the endosome membrane is considered a cell membrane. So maybe that's confusing you. You're thinking of only the plasma membrane. Yeah. All right. So we consider the endosome to be part of the cell membrane. When I say plasma membrane, then I mean the outer membrane of a cell. Okay. So if that's what's confusing you, that's good to know. Trimers, formation of trimers is a prerequisite for fusion, but that doesn't in itself expose the fusion peptide. All right? The fusion peptide is exposed in a trimer by low pH, for example. This is in the examples we showed. So just making a trimer doesn't expose the fusion peptide. And if you look back at the flu picture, you'll see the fusion peptide is at the bottom of the trimer. The only way the fusion peptide is exposed is when the pH drops, which is in a acidified endosome. All right, so membrane-enveloped viruses uncoat or release their genomes by fusing with the host cell, either at the plasma membrane in a neutral pH mediated mechanism or within an endosome. So conceptually, this is very easy. The membranes fuse and out comes the viral genome. Now, what about viruses like poliovirus here with a capsid? There's no membrane to fuse. How do you get the genome out of this very stable shell? So let's talk about that for a bit. So here's adenovirus, our virus icosahedral capsid with the spikes coming out of each five-fold axis of symmetry. So this virus proceeds through the endocytic pathway and is slowly dismantled. Really interesting. So the virus binds initially a fiber receptor. You remember earlier I showed you how the knob of the fiber sits into a cell receptor. That's the fiber receptor shown in red. Then the particle is endocytosed. The endocytosis requires a second receptor, an integrin shown in blue. So within a few minutes of binding the cell surface, the adenovirus particles get taken up by endocytosis. And then, as you know, as the endosomes move toward the cell interior, the endosome becomes acidified. And as that happens, one of the proteins embedded in the viral capsid, they're little yellow diamonds shown there. These are one of these accessory proteins that don't contribute to the symmetry of the capsid or the structure, but they have a function. That protein becomes activated, and it's called protein 6, and what it does is punch holes in the endosome membrane. So in the intact particle, it doesn't do that because it hasn't been activated by low pH. It's only when the virus is in an endosome that protein is activated. It punches holes in the endosome, and out comes the virus particle, which has been partially dismantled along the way. All right, so that's where the specificity comes from. It binds the right receptors and gets into the endocytic pathway, and then low pH activates this pore-forming protein. The virus particle then docks onto the microtubules to get the rest of the trip to the nuclear pore, and then the virus particle docks onto the nuclear pore complex and the DNA gets in. And we'll look at how that happens in a few moments. On the top right are some electron micrographs which show you some parts of this process. Here's an adenovirus particle moving down a microtubule. It's attached to a motor protein. That's the later steps here. And then the lower right micrograph are two adenoviruses bound to nuclear pores. So on the bottom of this picture is the cytoplasm of the cell. On the top is the nucleoplasm. Here's the nuclear membrane nuclear pores with adenoviruses bound to them, and that's the last stage of this uncoating. All right, so the answer for a icosahedral virus, one answer to getting it uncoated is to partially dismantle it and have it include a protein that gets it out of the endosome. And the complete uncoating, which happens at the nuclear pore, we'll consider in a moment. How about poliovirus? Since we have polio sitting here on the table, how do you dismantle it to get its genome out? And what happens is, now this is a entry that goes through endosomes, but low pH is not needed to get the RNA out of the particle. Here in the upper left is poliovirus binding to its cell receptor, and that is all that's needed to uncoat the particle. Binding to the cell receptor. You don't even need low pH. These particles are taken up very rapidly, and the RNA comes out of them. If you take poliovirus, purified poliovirus, and mix it with purified receptor, at 37 degrees, the RNA will pop out. So that's all you need. The receptor is in some way a catalyst for uncoating the particle. So how does it work? That's detailed on the lower panel of this figure. So on the left, you have a close-up view of the virus bound to two receptors. So let me orient you. Here's the interior of the particle. The RNA would be there. We don't see it in the first panel. But this is the five-fold axis of symmetry in the middle. And you see there are two receptors binding in that canyon that I showed you earlier in the slide around the five-fold axis. Now below the canyon floor, you can see here it looks like a spring. There's actually a lipid molecule that is present in the particle. If this here were a real poliovirus, this would contain 60 copies of this lipid. When the receptor sits into the canyon, it pushes the lipid out, and that's shown on the next slide. That gives the capsid room to move, if you will. It can now have conformational flexibility. And what happens is the proteins that are normally on the interior of the particle, these blue segments in the first panel, these are the N-termini of VP1. You could almost think of those as fusion peptides. They're very hydrophobic. When the lipid is popped out of the pocket by the receptor, those N-termini come out of the interior and they insert into the cell membrane, which is shown at the bottom, and they make a pore. And through that comes the viral RNA. So normally these hydrophobic regions are hidden in the interior of the virion. When the receptor sits down into the pocket, changes the conformation of the particle, the N-termini pop out, they make a pore in the membrane, and out goes the viral RNA. And interestingly, even though I'm showing here the 5' end coming out, very recently people have found that it's actually the 3' end of the RNA that comes out first. I don't know why that would be or how it's arranged so that the 3' end comes out first, or what provides the energy to do that. These are some of the mysteries that remain for understanding virus uncoating. I wanted to show you a picture of this lipid molecule that's underneath the receptor binding site. So here's a cartoon on the upper left of the five-fold axis of a poliovirus particle and a receptor sitting into the canyon. And just below the canyon, you can see the little groove, and that's where the lipid molecule is. In the structure of the virus shown below, you can see the cellular lipid in blue. So this is a subunit of the virus, one copy of VP1, 2, 3, and 4, and that's where the lipid would be. Now, many years ago, a series of antiviral compounds were developed to inhibit these kinds of viruses. They were useless clinically because resistance arose very rapidly and they weren't very effective. But they turned out to be critical for elucidating uncoating, because what these compounds do is they displace the lipid in this little pocket and they sit in there very tightly. So on the upper right is a model of how one of these compounds fits into this lipid binding pocket. So it has displaced the lipid and it's sitting in there. Now when these virus particles attach to a receptor, the chemical stays in there. Normally the lipid would come out and the virus could make a pore and uncoat. But when the antiviral is stuck in there, the pore cannot form and the virus never uncoats. So that's why these antivirals have antiviral activity. But as I said, they were never used clinically because they're not useful. On the bottom is a crystal structure of the entire virus particle mixed with one of these antiviral compounds. And you can see them in yellow. So there's one molecule binding at the base of the canyon. Again, to reorient you in the upper left, there's that little pocket where this molecule is binding. And how many binding sites for this chemical do you think are in the particle? If you remember icosahedral symmetry. You don't remember? Do you remember how many five-fold axes there are in a icosahedral particle? No. So there are 12 five-fold axes, and there are 60 subunits. So there are 60 binding sites for this compound, and that's what you can find in this diagram. Here's another cool example of why some viruses need two receptors to get into cells. Now, I showed you an example where adenovirus needs an attachment receptor and a second receptor for endocytosis. Not every protein on the cell surface gets taken up by the endocytic pathway. So apparently adenovirus binds one protein that just sits there, and then it binds a second protein to get it into the cell. Here's another example of this, which is even more cool. This is a Coxsackie virus entry. It needs two receptors. They're called decay accelerating factor, or DAF, and CAR. CAR stands for Coxsackie adenoreceptor because the CAR molecule is also required by adenovirus to bind. That's shown here. Now, both viruses initiate infection at epithelial surfaces, either the GI tract or the respiratory tract. And CAR is present in the tight junctions between cells. So here's a diagram of typical columnar epithelial cells that would be lining your mucosal surfaces. The decay accelerating factor is on the surface, but CAR is actually in between the cells. It's not accessible. The virus cannot bind to it. But Coxsackie needs it to get into cells. So what happens? So it turns out that Coxsackie viruses, shown in this slide in green, they initially bind DAF, which is on the cell surface. It's on the top of those columnar epithelial cells. DAF is present up here on the villus surface. The binding of Coxsackie to DAF initiates a signal transduction pathway, a series of phosphorylation events that have two consequences. First, they loosen up the actin filament network that's under the plasma membrane, because viruses can't move through that very readily, but this binding to the receptor loosens that up. And in addition, there's another signaling network illustrated here that ends up loosening the tight junctions. You know, these cells are very tightly held together by a variety of junction proteins. And the binding of virus to DAF on the cell surface loosens up the junction. The virus then surfs over to the junction, gets in, and binds to CAR, which is in here, and it can then enter the cell. How cool is that? So the virus needs two receptors, one to sit on the surface and tell the cell to move apart so that it can get into the receptor between the two cells. I want to tell you another example, because here we go back to reoviruses, which are the ones that have two icosahedral shells. They have an outer and an inner shell. And I told you that last time, and I promised to tell you why the virus has two shells. And here it's illustrated. And this is also the virus where the lysosome is needed for infectivity. So that's why I have to change that earlier question. So here on the upper left are reoviruses. Two icosahedral shells, an inner shell and an outer shell. It's a double-stranded segmented RNA genome on the inside. At each five-fold axis, there's also a kind of different configuration of protein. We call these turrets. And they pass from the inner shell through to the outer shell. So the virus attaches to a cell surface receptor. It's taken into the cell by the endocytic pathway. And so far, no changes. And then the endosomes fuse with lysosomes, which, as you know, contain many enzymes, nucleases, and proteases. And the enzymes in the lysosome digest away the outer shell of the particle. And you can see the change between the particle in the endosome versus the endosome-lysosome fusion. We call that a ISVP, infectious subviral particle. So this particle now is stripped of the outer shell. The inner shell, it turns out, is highly hydrophobic. So guess what? It can pop through the endosome membrane and get into the cytoplasm. And that this is now only the inner shell of the virus particle. We call it the core. And you can see the turrets still there. Those were present in the original particle. kind of obscured by the outer shell, but now they're very prominent because the outer shell is gone. So removing the outer shell is a consequence of the endosome fusing with the lysosome, and the enzymes in the lysosome do that, so that's very neat. The core is hydrophobic, so it can pop out of the lysosome. And then it's in the cytoplasm that this core begins to make mRNA. And in fact, the genome never comes out of that core. What comes out are mRNAs, and we'll talk about that next time. So that's why you need two concentric shells, and that's why you need lysosomes. All right, so this summarizes everything that we've talked about. Different pathways of entry. We've talked about fusion at the cell surface, which is pH independent. We have talked about uptake by the endocytic pathway, which is pH dependent. And there are different variations of endocytosis in cells. Not important for you to really know that, just that they exist. There's clathrin-dependent endocytosis, where the vesicles are coated with clathrin, this interesting triskelion protein. There's clathrin and caveolin-independent endocytosis. Basically, it's endocytosis, but there's no caveolin, there's no clathrin. And then there's caveolin-dependent endocytosis, which is really different. It's the same principle that you make vesicles and bring particles into the cell. But this kind of endocytosis targets the endoplasmic reticulum, which is really interesting. All right, so lots of endocytosis. And again, remember that these vesicles move through the cell by motor proteins transporting them on the microtubule network. And then eventually some viruses have to get in the nucleus. Not all viruses do, but some of them have to, and we'll talk about that in a moment. Most DNA viruses and a couple of RNA viruses need to get their nucleic acid into the nucleus in order to replicate. So I want to talk with you about that. And here are 4 different ways that this happens. On the left is the unusual RNA virus, influenza virus, that has to get its nucleic acid into the nucleus. Most of the RNA viruses stay in the cytoplasm. So if you remember our fusion discussion about influenza virus, after the virus and the endosome membranes fuse, the viral nucleocapsid protein is liberated into the cytoplasm, and it goes right through the nuclear pore complex. The nuclear pore can accommodate molecules up to a certain size, and the flu nucleoprotein can fit through it. And there's a whole transport process that brings molecules through the pore, and influenza viral proteins have the right nuclear import signals to do that. So this is very straightforward. But the DNA viruses typically don't fit through the nuclear pore. They're too big. So on the left, in panel B, we have herpes virus, which is way too big to fit through a pore, but it's got to get its DNA in the nucleus. So what it does, it docks onto the nuclear pore. And if you remember from last time, I told you, I made a point of telling you that one of the 5-fold axes of the particle is actually a portal. There's a hole in the particle. So the portal actually docks onto the nuclear pore, and the DNA comes out through the portal. The DNA is packaged under very high pressure in the particle, and it basically comes shooting out, although we don't know what the trigger is for that to occur. Now, in panel C is adenovirus, which you remember pops out of an endosome because it has a protein that's activated that then pokes a hole in the endosome. It's transported, it docks onto the nuclear pore. It's partially disassembled, and I'm going to show you in a minute the mechanism by which it's ripped apart to get its DNA into the nucleus. It's sort of similar to herpes, except herpes doesn't dismantle. Herpes has a portal through which the DNA comes, and adenovirus is dismantled. And on the far right, we have parvoviruses, which are, in fact, theoretically small enough to fit through the pore, but they don't. They bind to the nuclear pore, and they induce disruptions in the neighboring nuclear membrane, and the viruses go through that. How this happens, we're not sure, but we know that binding to the pore does that. It makes a hole in the adjacent nuclear membrane bilayer. The virus particles go in, and they release their DNA in the nucleus. Again, the trigger for that is not clear. So different ways of getting into the nucleus. Now I want to expound on the adenovirus because this is a new model that's arisen in the last few years, and it's pretty neat. So the virus is traveling on microtubules. It's left the endosome, and the partially dismantled virus particle is traveling down a microtubule. We know that the hexon proteins can bind both the kinesin and the dynein motors. Now the dynein motor, of course, brings you towards the nucleus. So that's what's happening here. The dynein is attached to the virus and the microtubule. It brings you down to the nucleus, and eventually the virus particle docks onto the nuclear pore complex, which is shown here at the bottom of the slide. then when the virus is docked on there, what we think happens is the kinesin molecules that are bound to the capsid engage the microtubule, and they start to pull the virus in the other direction, and that breaks it apart, as you can see here. So it's attached to the nuclear pore. It's also attached to a kinesin. Kinesin's trying to pull it back to the plasma membrane because that's what kinesins do. They go from the nucleus to the plasma membrane. The bidirectional torsion stress pulls apart the capsid, leaving the DNA. The DNA is shown here in blue. It's got proteins attached to it, which are then recognized by components of the nuclear import pathway. And the one shown here in this yellow sphere is called transportin. It recognizes the protein and imports the DNA into the nucleus. So the capsid is partially disassembled when it docks on the pore, and this pulling apart by the motors and the nuclear pore complex is thought to disrupt the capsid so the DNA can get in. So a variety of very interesting mechanisms for getting viruses into the nucleus. And next time we're going to start talking about what happens next. And the first thing we'll talk about is RNA synthesis.